Hello, my name is Jolene Ramsey. I'm a postdoc at the Center for Phage Technology in the Department of Biochemistry and Biophysics at Texas A&M University. Thank you for joining me today to hear about our team's work on using Galaxy and Apollo for bacteriophage annotation. The subject of our annotation efforts here at the CPT are bacteriophages, the viruses of bacteria. These are incredibly numerous, estimated to exist at about 10 to the 31st particles at any given time on the Earth, and that would be spread over all the different biospheres. Phage genomes usually, as you can see from this graph, are in the order of less than 200,000 bases in size, meaning that this is manageable for an individual to inspect and annotate to high quality. So both scientists and students at the undergraduate and graduate level will have an individual phage genome, newly sequenced, that they will need to annotate. We do our annotation at this time in a Galaxy instance we have at the CPT. This is a platform that will allow us to run typically command line tools in a web browser interface. And this is extremely powerful for all of our computational analyses. We equip individuals to perform self-paced analyses by providing tutorials and frequently asked questions on our Galaxy Training Network format website. And we also, in the context of a formal course at Texas A&M, provide training in phage biology to allow individuals to be equipped to completely and fully annotate high-quality genomes for deposition into the databases. Not all of our analyses, though, are performed in Galaxy. After we run tools, we then utilize the very powerful collaborative genome editor Apollo, which we also run an instance at the CPT, which allows for us to analyze contextualized data and make informed decisions about how to predict what is present in these phage genomes, and this we can do together as a team. After going back and forth between Galaxy and Apollo, we can then take these formatted files and put them into either public or local databases where we can deposit our curated data for the scientific community to have access to. At Texas A&M, I mentioned we have a formal course called Bacteriophage Genomics, typically offered to upper-level undergraduates in the spring of every year. And this began with traditional lectures on phage biology and transitions into the computer lab section where students will take a phage genome and run it through our pipelines to annotate it by the end of the semester. This course also includes a lab component where they isolate a novel phage from the environment, resulting in 3 student-owned products coming out of the course: the GenBank file of their annotated phage genome, which will be put into NCBI, the manuscript that they write on their annotation that can be submitted as a genome announcement, as well as the novel phage they've isolated from the environment. We'll be focusing here on the bioinformatic pipeline aspect of what is taught in this course, and that centers on 3 main workflows, the first of which is the structural workflow, where the features of the genome are called, primarily the genes, as well as terminators and other features. That is followed by the functional workflow, where protein function is predicted using both sequence properties as well as similarity to known proteins through familiar tools including BLAST and other conserved domain predictors. And in parallel, we do comparative phage genomics in the comparative workflow to look at relationships with other known phages at both the nucleotide and amino acid levels. All of these systems are available at our website, cpt.tamu.edu, and I will briefly walk you through what each of them looks like, starting first with our course tutorials, which is in the Galaxy Training Network format and constantly being updated. In its current form, we have 4 primary topics. Those include Introduction to Galaxy and Apollo, how to assemble novel genomes, as well as a section on the phage annotation pipeline for each of the different workflows, and several different analyses, including how to add custom evidence tracks, finding lysis genes, or reopening a genome. Now, our entire pipeline has been described in a manuscript that is currently under review and should be available shortly. This describes the establishment, development, and details of current running of the system. The way it works is you input a newly assembled or empty genome into CPT Phage Galaxy. We describe and teach how to navigate through the Galaxy system, including the use of the history for tracking input and output data, searching tools in the left panel, both internal tools that we have developed for phage annotation needs, as well as external tools that we have added developed by others, and navigation of the center pane for setting parameters for individual tool jobs, as well as complete workflows. Then, in the bridge between Galaxy and Apollo that was originally developed by Helena here at the CPT, we exchange data and move this information into Apollo, where we help individuals navigate through the use of Apollo, including the turning on and off of annotation tracks shown here on the left. These are evidence that is displayed underneath its corresponding source location on the genome, shown in the yellow user-created annotations tract. The predictions can then be made in the context of all of that evidence, and this can be done together as a team in a collaborative, real-time fashion. After the analyses are complete, these are readily available to be exported and provided to the scientific community through public means. We start with the Galaxy structural workflow. So the phage genome is the only input data set for this, and there are 4 major tracks of tools here. So each box shown here is an individual tool for which the parameters can be individually set, and these are interconnected to provide the input and output for the following tools in the workflow. You can see here we've got a tRNA calling track, as well as 3 separate gene calling tracks that will all be fed into a JBrowse instance that is then loaded into Apollo for further viewing and analysis. The 23 steps in this workflow typically take about 15 minutes to perform, and those are then available for view in Apollo. This is what an empty genome record will look like. No user-created annotations. Our users become very familiar with navigating both at this level, as well as zooming into sequence level and turning on and off the evidence tracks shown here at the left. The recommended order of operations for analyzing the structural information is first to call tRNAs, then protein coding genes, and last terminators. With protein coding genes, there are 3 gene callers that we use, and this is to allow us to get proper starts for every gene, and we are able to analyze the best way to get maximum coverage of coding sequences in the genome, as well as checking orientation and potential insertions such as morons. When you turn on all 3 gene callers, such as you see here, where you have the light blue glimmer track in light blue, the metagene annotator track in dark blue, and the naive ORF caller 6-pack in green, you see there's lots of information. What we have added in particular is Schindel-Garno sites, which are important for translation to every predicted coding sequence that has them. And these can be taken together. Sometimes it's very easy. This could almost be done in an automated fashion where they all agree. Often, though, you'll see that here there are disagreements where this is the call that we eventually decided on, but Glimmer had a different option, and 6-pack had actually 3 available options. So the appropriate choice was made by just right-clicking on the preferred evidence and calling the gene. And you can see in this case where the preferred gene callers actually did not have the complete open reading frame. Instead, the naive ORF caller did, and we were able to confirm this by looking at additional evidence from the functional prediction. After gene calls are completed, this is what's necessary for the start of the functional workflow, where we predict protein function. This looks at sequence properties as well as similar to known proteins in using tools such as the familiar BLAST or conserved domains like transmembrane domains, and potentially comparisons to proteins with known structure. So this requires the gene calls coming out of Apollo and being ported back to Galaxy through our bridge, and then the running of the functional workflow, which has 57 different steps. I don't expect you to be able to see that here. We have tracks for finding lysis genes, looking for potential intron coding sequences, as well as lots of different BLAST analyses and conserved domain prediction. The time to complete this particular workflow varies based on the input genome size, and so this is on the order of hours to sometimes just left overnight. After all of the functional prediction tools have been completed and the JBrowse instance is finished, those functional predictions are given back to Apollo, where they can be displayed in the dynamic viewing interface of Apollo. You can choose which evidence tracks to display here below the gene calls. And the typical order of operations recommended for the functional workflow is to first look at the BLAST results for both BLAST-P and BLAST-N. We have both external databases as well as local databases. Here, the first one, Canonical Phages, is our local curated database of phages that are known to have experiments performed on them in the lab, in which we weight as a higher quality evidence for a potential functional prediction, followed by externally curated databases like Swissprot, and finally catch-all databases like the NR from NCBI. After looking at BLAST, we recommend looking at conserved domains, the results from InterProScan or other transmembrane domain callers. And then finally, we have tutorials that are available for looking at all of the tricky features like finding introns, inteins, frame shifts, and lysis genes. In parallel with the functional workflow comes the comparative workflow, where we will look at phage relationships. This also only requires gene calls coming out of Apollo through the bridge into Galaxy. The comparative workflow that we have set up is only 11 steps and typically takes less than 20 minutes to complete. It analyzes, it compares to other phages, both at the protein level as well as at the nucleotide level, and comes out with 3 primary outputs. That is a protein similarity table where you can see the name and accession of the phage, as well as how many proteins are similar between the 2 phages by BLAST. There is a genome dot plot that is given to see at the whole nucleotide level what the similarity is between the phages being compared, as well as a final nucleotide similarity calculation that is given. We have additional tools available beyond the ones that are in the workflow in our Phage Galaxy, and those include things like phage term, which will help you to predict the termini of the novel phage, as well as a variety of tools for drawing genome maps, such as this comparison map here shown from EasyPhage. That completes my brief overview of our annotation system for scientists and students to do phage annotation through the CPT Phage Galaxy and CPT Apollo. Using the contextualized evidence and the back and forth that's able to be performed individually and as a team, supported by tutorials and training in basic phage biology, for us to contribute this annotated information back to the scientific community. All of what I described is freely available to the public on our website, so hope to see you there. With that, I would like to thank all of the team at the Center for Phage Technology, our PIs Rai, Jason, Jim, and our director May. The systems admin that originally started this project was Helena, and our current system admin Corey. Our programmers over time have been Eleni, Anthony, and Curtis. There have been many, many CPT Lab and TAMU users who have tested the system, and many, many, many students who have gone through our bacteriophage genomics course. I'd also like to thank the Apollo team, especially Susie Lewis and Nathan Dunn, for helping us to adapt it to phage annotation, and the Galaxy project, the entire Galaxy community, as well as the Galaxy Training Network for the tutorial formatting. The Center for Phage Technology receives funding for this project from the National Science Foundation and also from Texas A&M AgriLife Research and from Texas A&M University. You can reach us for questions if you don't make it through this conference at cpt.tamu.edu or on Twitter. Thank you for your attention.